All right, so we've got our fatty acids into the cell. And if we're in a need state, we're going to be breaking those down via beta oxidation in the mitochondria. But free fatty acids sitting in the cytosol of the cell aren't ready to do that oxidation in the mitochondria. So two things that we have to do first before we're ready for the steps of beta oxidation is we need to activate those fatty acids so they're ready to be broken down, and then we need to transport them into the mitochondria. Remember, the rate determining step of fatty acid breakdown is the influx of those fatty acids into the mitochondria. So we're going to address and talk about how that happens. So this slide is going to address two main things. We're going to talk about this activation piece. Okay, and this activation piece is really what we're seeing right here. So we're going to talk about the details of what we see here in just a minute, but this is our activation piece right here. And then we're going to be talking about our transport piece. And our transport piece is what we see here. Okay, so first, let's talk about this activation piece. So key here, and I'm going to talk about this generally before we kind of look at some structures and talk about some specifics, is if we look at the functional groups that we have on a carboxylic acid or on a fatty acid, we basically just have a carboxylic acid group, and that's not really a good functional group to be doing chemistry. So what we're going to be doing is we're going to be adding it on to CoA. Okay, and adding it onto CoA is going to generate a fatty acyl, CoA, and that's going to be a better functional group to do some chemistry. So we're going to have a fatty acyl, CoA. And I'm going to talk about two different hats that we're going to put on here. And really, the first thing that we're doing here is we are putting on our CoA hat. So our CoA hat, we're going to draw like a little blue derby hat. And so we've got a fatty acid that now has this blue derby hat on. Okay, so that's the details in kind of, or in a simplistic way of what happens during activation. So let's zoom in here and kind of look at some structures and talk about some specifics of this. Okay, so we highlighted that when we have a fatty acid, there is not a good functional group that we have on fatty acids for doing chemistry. Okay, so what we're going to do is we're going to append it to CoA, and that's going to allow us to then do some additional chemistry with that. So we can't just like take something that is not a good leaving group and couple, or that is not a good functional group, and just kind of have it be coupled without the input of energy. So here's how this process is going to occur. Okay, we need to use a little energy, okay, a little biological money, okay, and we can see here a little bit of mechanistic drawing here. We can see nucleophilic attack by that fatty acid onto ATP. We're going to be displacing pyrophosphate here. So this is an important piece that we've talked about before. So here again is pyrophosphate. So pyrophosphate here. And what we've done is we have added an OAMP group, okay? And the important part of having that pyrophosphate as a leaving group is that imparts irreversibility. Okay, so we are going to have irreversibility. So anytime we see the generation of pyrophosphate, because we've got this inorganic pyrophosphatase that works in an irreversible way to generate this phosphate, two phosphate groups, this sort of pulls this reaction forward. So even if this was a reversible process, by removing that pyrophosphate, it imparts that irreversibility. Okay, so the thing that we've done here is we've added this OAMP group. So that was the intent of having this, is we've got this OAMP group. Okay, we've seen this before when we did our reaction mechanisms, we saw this. Oftentimes, we are going to activate a molecule towards doing some coupling because we're going to add a nucleotide derivative. Okay, in this case, it was using ATP to add an OAMP group, and that's important because this is a good leaving group. Okay, second sort of chemistry that we have here, here's our blue derby hat. Okay, here's CoA. We are now going to have nucleophilic attack displace this good leaving group, and we generate our fatty acyl CoA. Okay, so we needed to have some money for that, but the net is that we have taken our fatty acid that had no good functional group and we've put it onto CoA, so now we've got the ability to do some chemistry. Okay, so now we need to transport this into the mitochondria. So I hope I've convinced you that because the rate controlling step of fatty acid breakdown has to do with whether or not those fats just get into the mitochondrion, we're not going to have that happen super easily. It has to be something that we're making sure that we want it to happen. Okay, so again, fatty acid breakdown happens in the mitochondria, and the influx of those fatty acids into the mitochondria is the rate controlling step. So highlighting importance here, we've got fatty acid breakdown. It's going to happen in the mitochondria, and this is the rate determining step of fatty acid breakdown, is whether or not those fatty acids get in there. Okay, so highlighting and just sort of connecting these pieces together, here is our fatty acyl CoA right there. Okay, so what we're going to do, and I'm going to highlight here just with another color, we've got our fatty acyl portion. Okay, let's highlight our fatty acyl portion that we've got right here. Okay, that is going to be the same as we have down there. So what we're really going to be doing is changing the hat that we have. Okay, so let's kind of walk through this sort of before we look at the picture, just with some generalized sort of structures. Okay, we're going to have, in this case, a trans-thioesterification reaction. So if you have a thioester, for example, like acetyl-CoA, and you have another alcohol group, or it could be a thiol group here, but in this case we're going to have an alcohol group, we're going to do trans-thioesterification. Remember, an ester is a combination of a carboxylic acid and an alcohol. You can swap out what that alcohol group is by doing a trans-esterification reaction. Well, if it's a thioester, then you can do a trans-thioesterification reaction. And what we're really going to be doing is we're swapping out, okay, now we're going to have this other alcohol group there, and we're going to be left with our free thio. Okay, so what we're going to have here, and I'm going to sort of write it down on the bottom here. Well, actually, let's run through the picture first. Let's run through the picture. So what we're going to have here is this OH group that we have right here, okay, is going to be this guy right here. So this is going to be what we call our carnitine hat. So this carnitine guy, we're going to draw as kind of a red hat like this, okay? So that's what this guy is. It's a little red hat. So let's look at the picture, and then we'll sort of summarize both pieces, kind of an equation down below. So let's take a look at this. So here, we're in the cytosol. We've activated our fatty acid by putting it on CoA. So we've given it that blue derby hat, and now it has some functional groups that allow us to do chemistry. We're going to do this trans-thioesterification reaction where we're going to have carnitine and the red and the blue derby hat kind of swap spots. Okay, so in this case, we're going to get free CoA, so the derby hat comes off, and then we are going to attach that red hat to our acyl group. So again, here's our acyl group, and now we've got our fatty acid wearing a carnitine hat. Now, why is this important? Because there is a transporter that recognizes things that have carnitine on them. So if you are wearing a carnitine hat, you can go through this special transporter. So we can take this fatty acid that now is wearing a carnitine hat, it can go into the mitochondria, and then inside the mitochondria is going to be another version of its blue derby hat. So there's going to be CoA that we have there. So it's going to take off its carnitine hat. We're going to get free carnitine back, and then we are going to get our fatty acyl CoA, And as soon as this guy gets into the mitochondrion, we are ready to do fat breakdown. So again, the rate determining step is fatty acyl-CoA in the mitochondria. So if we've got that guy in there. All right, so let's sum that up here then with kind of down here at the bottom. So let's go ahead. I'm going to zoom in down here just so we can kind of put this all together. So we've got a fatty acid that's in the cytosol that basically has no good functional groups. So we are going to be doing activation chemistry. So that's what we had up above in blue. It's going to take a little money for that. So we need ATP, and then we need CoA. So that was our blue derby hat. Okay, so both of those are required pieces, and we are left with fatty acyl-CoA. And again, this is in the cytosol, so our fatty acid is wearing its blue derby hat. Okay, now what's going to happen is we are going to be doing a trans-thioesterification reaction, and we're going to swap out the hat that we have. So putting on this red hat is going to be happening because we've got trans-thioesterification. Okay, I'm going to go up top here just so I have a little bit more room. Okay, and we're going to have our fatty acyl-carnitine molecule. So again, putting on that red hat, but we are still in the cytosol. But now, because we are wearing that red hat, we can go through that transporter. So even though there's multiple parts that are part of sort of this transport system, it really is about putting on that carnitine hat so that we can take and transport those into the mitochondria. And then we're going to have fatty acyl-carnitine that is now in the mitochondria. Okay, and now what we need to do is we need to do another trans-thioesterification reaction, this time taking off our carnitine hat and putting back on our blue derby hat. So reminding us this is another trans-thioesterification reaction. And now we're going to have fatty acyl-CoA, so back on with our blue derby hat, and then we are in the mitochondria. And so this means, very importantly, we are ready to do beta oxidation. And beta oxidation is the process whereby fatty acids that are in the mitochondria are broken down into acetyl-CoA, which can ultimately be used for energy.